Human Genome Project. Now, what is this Human Genome Project? It is a mega project which was actually constituted or established in the year 1989. And before going to the Human Genome Project, HGP, what is a genome? Human we know, project we know commonly, the meaning of that word. Genome is the total number of the genes, the sum total of the genes present in the haploid set of chromosomes. So that means in human being, we are studying about human. So in human there are 46 chromosomes, 23 pairs. So out of these 23 pairs, when we remove one chromosome from each pair and separate, so that set of chromosomes, 23 chromosomes, is called as haploid set. So the sum total of the genes present in the haploid set of chromosomes is called as genome. So what genes are present in one set, similar complementary genes are present in the other set. So whenever we want to study the genes of a cell, human cell, we need not study all the genes in the 46 chromosomes. When we study the genes of one haploid set, it is we can come to know the other type of genes in the other haploid set. Now in the year 1989, so this project was constituted by scientists of different countries. So this is just an introduction to this project. Now this committee or the group which was constituted, it was headed by James Watson, what we study about the double helix of this DNA. So James Watson was the head. He led the committee or the group which was conducting the Human Genome Project. And they decided to start the project in the year 1990. So that means next year, 1989, they decided. But they also decided to start the project from 1990, and it should be completed within a period of 15 years. And also it should be completed within an estimated cost of about $3 billion. So that means by 2005, this project should be completed. So that was the decision taken by the group of that project. Now, as per their functioning, so they started, and 99% of the work was completed in the year 2003. Only 1% of the project was left. So every chromosome they studied. So the chromosomes are given a number, first pair, second pair, third pair, fourth pair, like that. So only the chromosome number 1 was left to be studied. So remaining all the 22 chromosomes were studied. So 99% of the work was completed in the 2003, and again by 2005 the remaining small percentage was completed, and the Human Genome Project was successfully completed. So by what is the use of studying this genome? So by studying the genes, by studying the genome of an individual, we come to know what characters are present in that individual. So what are the actually characters? So the certain characters, phenotype, what we say, they are not genetically. So we cannot expect that whenever there is a morphological expression or whenever there is an external expression, so that is the genetic one. So but by studying the genome, we come to know what actually the genes are there, what are the actual characters present in that individual. That is why the Human Genome Project was very useful. And what are the salient features of this project? So that we have to study. So the salient features, which is usually asked for 5 marks. So any 5 of these points we have to mention. So the genome which is present in the cell, it contains this much of DNA length, 3164.7 million nucleotide sequence in the DNA. So all the chromosomes, 23 chromosomes, genome, it is having this length of DNA, about 3164.7 million DNA nucleotide bases, that is the length. And after the studying the genome, it was also found that there are about 30,000 genes present in our genome. So that means we are having so many number of characters in our body. But however, when we go on making the list, we can mention only some few hundreds or like that. But there are other certain internal characters which are related to our metabolic activities, which we cannot see them morphologically, or they are going on in their biochemical reactions. So that is why when we go on studying the genes or the characters, so it was found that there are about 30,000 genes in our genome. That was known by this project. Then what is the length of a gene? So this is the length of the DNA. So in this amount of DNA there are 30,000 genes. So however, all genes are not equal, but an average gene is having 3,000 bases. So there are certain genes which are lesser than 3,000, but there are some genes which are more than 3,000. But when we take at the average number, so a gene is having a length of about 3,000 base pairs or the nucleotide bases. And it was found that the largest gene in our body was the dystrophin gene, the gene which produces the protein dystrophin, which was having the largest length, about 2.4 million nucleotide bases. So that is the largest. So that means all other genes are lesser than this length, consisting of few thousands, some maybe more than 3,000 and some less than 3,000. So average is about 3,000 nucleotide bases. And also it was found that the DNA present in the individuals, in all human beings, irrespective of age, whether it is a child, middle age, old age, or a male, female, like that, it was found that 99% of the nucleotide bases were same in all individuals. So that means the DNA of about 99.9% in one individual is same in the other individual. But we are differing only in about 0.1 percentage. So we are differing in that minute percentage of DNA. So that we came to know only after the studying the human genome project. And then out of the discovered genes, 50% or more than 50% of the genes which were discovered, they were not exactly, the functions were not known. However, the occurrence of the genes was identified, but the function of each and every gene was not known. But nearly 50% of the genes, we are knowing them, the functions of the genes. But the remaining 50%, more than 50%, their exact functions is not known. So that is yet to be discovered or in the future. Then out of this total genes in the genome, less than 2% they code for the proteins. In other words, they are involved in the protein synthesis. So less than 2% of the genome, the genes of the genome, they code for the proteins, and that is another important character of this human genome project. And this genome, it also consists of a large portion of a repeated sequence of DNA. So however, this repeated sequence of DNA, it is having different peaks which are called as, and that we will be studying in the DNA fingerprinting. So after studying the genome, it was found that there was a large portion of the repeated sequence of the DNA in the genome. So that is also another important feature we obtained from the HGP or the Human Genome Project. And out of the total chromosomes, the chromosome number 1, which was last studied, so which I just mentioned, 99% of the project was completed in 2003. So the remaining 1% was actually this one. So chromosome number 1 was very difficult to study, because the reason was the chromosome number 1 had maximum number of genes, 2968 genes were present. So that means in the other chromosomes the genes are lesser than this number. And the Y chromosome of the male has the least number of genes, about only 231. However, Y chromosome is not present in females, only there are two X chromosomes. So Y chromosome is having the least number of genes. Chromosome number 1 is having the maximum number of genes. So that we obtained this point we obtained from the studying this human genome project. So this human genome project has helped a lot extensively, especially in medical research or even in case of pedigree analysis. So when we study the genome, we come to know what disorders are present in that family or in that individual, and thus it also helps in the pedigree analysis of the family. So that is about the human genome project. So these are the silent features. So thank you.